Hey biology, happy Thursday. I do have an animal for you, but I'm going to do that on a separate post. So that's coming too. All right. But this post is just about what needs to be done for biology. Okay. First off, your grades are posted. Your third quarter grades are posted. So we are moving on to the fourth quarter. And some of the first stuff that's going to be in your grade book is the ed puzzle and the quizzes, which almost all of you did. Yay, good work. Okay. And so you're starting off strong. Those were both kind of like review things, because guess what? There is a test in your future. Okay. It's going to be an open note test, because that's just what we can do, I guess. But I think organizing and making sure you can do some of the skills for the test will be important to be able to do well. Okay. So first, and hopefully you have organized this already because you used it on like the crossword question review, but make sure you have your 3 sets of notes: reproduction and basic heredity, patterns of heredity, and protein synthesis. Okay. If you are missing any of those 3, I can send you a copy of the notes. Okay. So just tell me, email me. Now, you will also need the codon chart. You will be doing some transcribing and translating, going from DNA to mRNA to amino acid sequence, okay? So make sure you understand how to do that. Maybe I'll just, and that's where you go, right? It seemed like most of you did it on your review, but a few people left it blank. So again, it should be on the back of your protein synthesis notes, but it has all the DNA bases and what they go to. So if we're going DNA to mRNA, A goes to U, T goes to A, C goes to G, and G goes to C. Then we read those mRNA bases in groups of 3 called codons. Okay, and you use your codon chart, right? First base on the left, A. Second base on top, U. Okay, we go to where those 2 boxes come together, and then the third one is maybe A, and we come across from there. Leucine, I don't know, I just made that up, but whatever one it is, okay? And it is totally acceptable to abbreviate amino acids by the first 3 letters, okay? So you can do that on your test if you'd like to keep things a little shorter. All right, a few other things that will be on there are your Punnett squares, okay? So let's just talk about Punnett squares a little bit. So first off, the different kinds of dominants that we have talked about so far in this section are complete dominants. That is where the heterozygote looks like the dominant allele. So that's like your basic dominant situation. Okay, if nothing is explained in the question, you assume that it is complete dominance. Okay. Now, sometimes the heterozygote looks different. If the heterozygote looks different than the homozygous dominant and the homozygous recessive, then it is either codominance or incomplete dominance. Okay? Codominance, the heterozygote expresses both. AB blood is the classic example. Okay? But spots could be a roan horse, all right? Incomplete dominance, the heterozygote is a blend, all right? And so if you have like black, white, heterozygote would be gray. Curly hair, straight hair, heterozygote would be wavy. And if the heterozygote is different, it will be explained in the information, except for blood type. I'm expecting you to have that memorized. Okay, you just know that's codominance. And the last one was sex-linked. And those were traits that are carried on the X chromosome. So they are inherited differently in males and females because males only have 1 chance to get the allele because Y is too tiny and only carries maleness. Okay, color blindness is the classic example of that. All right, so I'm going to do a few examples. I'm going to ask a question And then I would suggest you do it at home. Like pause the video, do it on a little scratch piece of paper, and then I'm going to show you the answer and you can check and see how you did. All right? So let's just say to start, we had, let's say free earlobes are dominant to attached. So I'm going to use a big F for free earlobes. And let's say dad has free earlobes, okay, but his mom had attached. Okay, and we're going to cross that with a mother who has attached. So the genotype of the parents should be as follows: heterozygote times the homozygous recessive. All right, quickly, this is an easy one, right? We're starting off easy. Set up a Punnett square for that cross, and then tell me what percentage will end up with free earlobes. Okay, so go ahead and pause it, do that, come back, and I'll have mine up. Does yours look like this? Remember, it doesn't matter which parent is on which side, so you might have those switch, but you should have 2 heterozygotes for free earlobes and 2 homozygous recessive attached. All right, let's step it up. Okay, let's do some blood typing. Mom has O blood. Dad has AB blood. He's the co-dominant. Okay, so the parents are, you got something like that? Okay, remember O is recessive. All right, pause, do your Punnett square. All right, so what percentage of the offspring will have O blood like mom? What percentage will have AB like dad? And then is there any that got A or B even though mom or dad wasn't A or B? Is that what you got? Of course you did. Nice work. All right. So let's try one for incomplete dominance. So I'm going to tell you that red and white are incomplete dominant. Okay? So if we cross a mom who is red and a dad who is white, say these are flowers, what percentage of their offspring are going to be red? Did you get that? Nice. All right. Sex-linked. Okay, for whatever reason, you guys love sex-linked. It seems like I always get tests that somebody does everything sex-linked. Okay? Don't do it. Only do sex-linked when it says it is sex-linked. Okay? And so the ones we've been talking about the most are color blindness, hemophilia, and calico cats. All right? So remember, whenever you do sex-linked, first you're going to have to set up the sex chromosomes, right? Mom is XX, dad is XY. Okay, let's do this for hemophilia, a recessive disease where your blood doesn't clot. Okay, and let's say dad has hemophilia. Okay, and mom does not, and she does not carry hemophilia. Okay, she has normal blood. All right, So mom and dad's genotype should look like this. Okay, go ahead and do the cross. So pause, finish up your cross. All right, so it is important to maybe make your boxes a little bit bigger with sex linked, especially with my Sharpie. They got a little squished, okay? But what you should see is that both males inherited the normal allele from their mom and then Y from their dad that made them a boy, right? And then each of the females, that way, do not have hemophilia, but they are carriers, okay? They carry that allele from dad. All right, so how many of the offspring have hemophilia? 0, none. All right, last one, just to make sure you're pro. Okay, calico cats was a really interesting one because it's sex-linked and codominant. Woo! All right, so we are gonna have, we'll do it the same as the pink packet. We had R for orange. That was kind of weird. And then we had B for black. And that's the alleles. They are codominant, so they're both big. If an individual ended up with B and R, they were both orange and black, calico, okay? Now this was sex-linked as well. So I would like you to do a cross between a calico female, love calico cats, and let's say an orange dad. Actually, I really like orange cats too. All right, so go ahead and do that cross. Remember, we're doing both sex-linked and codominant. Go ahead and pause and finish that. All right, so how many were calico? How many were black? And how many were orange? Was your cross something like this? Okay, I got one calico female, one orange female, one black male, and one orange male. Okay? All right, if you have questions or you want me to go over anything tomorrow before we hand out the test, please leave a comment below or email me. Email is a little more reliable. All right, so a few of you haven't turned in your review yet. If you haven't, make sure that gets in and get organized and ready for your test tomorrow. And I'll give you a couple days to do it. Okay? All right, have a great day. Bye.